We're not selling them, even though we get so many kids and parents calling us, begging us to sell them. We're not doing that. I do think other than the cold tolerant experiments, the other thing that we'll probably do is if there are additional traits that we want to test from our Agent Elephant editing work in Woolly Mouse 2.0, we'll probably do that.
Hi, this is David Crow with Den of Geek, and I am here with the CEO, Ben Lamm of Colossal Biosciences. Good afternoon.
Thanks so much for having me.
We don't normally do traditional science interviews, but we also don't normally have the offer to speak to someone in the CRISPR and gene editing space. So y'all have had a big week, and I thought a good place to begin is maybe tell us what y'all have been up to as of late.
I think that we didn't mean to, but we kind of accidentally broke the internet with the woolly mouse. We thought they were, pretty interesting from a proof of concept perspective and verification of our edits that we've been working on for the last 3 years for the woolly mammoth. But then they had this massive adorability factor that we didn't plan for, which also gives us a lot of hope for our first mammoth calves will also be this adorable. And it's been crazy. We've just had lots of great folks like you that we've been talking to. We've also had, parents and kids calling us and emailing us and DMing us and just being like, can we buy one? Like, what does this mean? Is this a mini mammoth? We've got all these questions. And it's kind of like spurring, it's had this like weird halo effect of like spurring additional interest in science.
I don't know if you planned it, but you may have opened a merchandise vein that you could tap into with this.
Yeah, we're not selling them. I've actually seen on X, people have made shirts. I think that there's already a meme going. It's kind of gone a little weird really, really quick, but people are excited, which, if we can do things in today's age that gets people excited about science and conservation, then that's a win.
Obviously, there are countless extinct species to research, to study. What about the woolly mammoth was the one that you and geneticist George Church chose to pinpoint to advance this technology? Well,
so George, and it probably would have been better if we just started with like the easiest thing in the world, but we're like, what is the hardest, most complicated thing that we could start with? And we're like, elephants. No, but George had been working on this for 6 years before I met him, right? So he'd been doing the computational analysis, and he had thought that, we do have the technologies with enough funding and focus to bring back the mammoths. There's an ecological benefit to putting them back into the Arctic, which is pretty cool. And then there's this halo effect of we're not really doing enough to save elephants. And there's a lot of ways we can innovate to save species, specifically elephants. So George is very passionate about elephants. And so I guess that together was like the right thing. And then I was partnering with George, needed his technology, needed his innovation. And so the mammoth was kind of our big flagship project that we started with.
Could you talk a little bit about how you were able to verify, what woolly mammoth hair looked like 12, 15, 50,000 years ago?
So we actually know what mammoth hair looks like because we pull it from the permafrost. In our new lab, we actually have this Peter Jackson's one of our investors. We're pretty close with Peter, and Weta Workshop actually built this huge 12-foot mammoth of a mammoth suspended in ice, because I think that's how people think about it, but it's actually much grosser from what gets pulled from the permafrost. But when you actually take mammoths, Siberian mammoths or Alaskan mammoths out of the permafrost, and if you do have tissue samples or hair and it gets cleaned off, it is kind of this reddish brown golden fur. It's actually quite beautiful, and that was one of the targets in the mice that we were hoping to get with our phenotype engineering, and we were successful, which is pretty cool.
So the hair and for that matter the body fat composition, it matches what a woolly mammoth would be.
Right. So we had 3 kind of big targets. We actually made 8 edits across 7 genes using precision gene editing. So it wasn't like we were just with CRISPR knockouts, and sometimes you have off-target effects. It doesn't always work perfect. You have unintended consequences. We spent a lot of time in the last few years actually optimizing our pipeline so that we can quickly deploy the edits into a cell. And then we have a screening process, so we only bring cells to life that are healthy. So all the mice that were born, all 38 of them, were all healthy. They all had the edits, and we had 100% editing efficiency with 0 off-target effects, and we delivered it all in one goal, which is called one delivery, which is called multiplex editing. So what's crazy about the woolly mice is they're one of the most genetically modified organisms on the planet, and they're probably the most precision targeted genetically edited animal on the planet, even those little guys. And so we did fat metabolism for mammoths, we did hair color, and then we did that wavy, curly, or as our chief science officer likes to say, floofy fur, which I don't think is a scientific term. But yeah, that was those were kind of the 3 target traits we were looking for.
And could these mice survive in a tundra-like environment in Siberia?
Yeah, so they are a little bit heavier, right? But they're still kind of within the statistical range of what mice typically are. Obviously, their fur is much thicker, so they look bigger. But from size-wise, they're about the same. We probably need about 6 to 12 months to verify cold tolerance. We have an ethics board and we have an IACUC board, which actually looks at all the experiments that we submit to see if we can do them, and making sure that we're only doing behavioral studies that are healthy for the animals. We're not going to just take them and drop them in the Arctic and see what happens, right? It's a very thoughtful process compared to other behavioral work that we're doing specifically with existing kind of wild type mice. And then that comparison, that'll probably take 6 to 12 months. And then we're going to update our scientific paper with the answers.
Well, that is very exciting, really. And but obviously a pivot point for an outlet like Den of Geek is going to be, you've seen Jurassic Park, yes?
I have. I actually wore a shirt that said preoccupied today because, believe it or not, we've actually heard that once or twice. We do get the question a lot, like, didn't you see Jurassic, or don't you know how Jurassic Park ended? And I was like, we also know how Dune ended, and Blade Runner, and a bunch of other fake sci-fi movies that were designed to entertain us, right? Unless I missed it, I don't think Jurassic Park really had a conservation narrative to it. They weren't trying to help species like we are, because we make all of our tech available for free for conservation. And I don't think that if the movie was real, which it wasn't, I don't think that they were doing it to inspire kids. I think they were doing it to get people excited and come to the park.
Were you inspired to go into biosciences specifically because of this movie?
I think all sci-fi has inspired me to be in tech. You know, I'm not a biologist. I'm not from this world. I just like to build teams of people that are much smarter than me. And I just got really excited about the idea that Jurassic Park-like technology was possible. And it also had this halo effect of helping existing species. So it's like we could create value, inspire people, and, you know, also make an impact. That kind of like Venn diagram got me pretty stoked when George pitched me it.
Now that you are in that the gene editing space, do you go back and watch the movie, and are you like, well, they got some good
we're almost doing like reverse Jurassic Park, right? Because they were taking amber and taking DNA from dinosaurs and filling it in with a bunch of stuff. We're doing exactly opposite, right? So we're starting with an aged elephant cell and then editing it with specific mammoth alleles that we found. So we're doing it a little bit more precise than they did it. And also, not that we've tried, but amber is not the best DNA store. And so there is no ancient DNA. So if that question's coming, can you bring back a dinosaur? Whether you're, depending on how you feel about it, sadly no, or excitingly yes, or excitedly no.
Obviously not amber, but there's no other avenues you've considered exploring.
So we get this question a lot. So right now, our oldest genome we're working with on the mammoth is 1.2 million years old. So it's pretty old. But dinosaur DNA is 65 million years old if it were to exist. So you can demineralize dinosaur bones. One of our scientific advisors, Kenneth Lacavara, did this, which is pretty cool. And you can identify specific amino acids. And so you could kind of like figure out, is this a triceratops from like a bone if you demineralize it and run these tests? But we're talking about fragments of a fragment of fragment. There's not enough DNA to ever rebuild something.
In the science space, in the tech space, do you think sci-fi is like a source of inspiration for a lot of people in this line of work?
Yeah, I think it should be, right? It's like a lot of these incredible comic book artists, graphic novel artists, and movie producers and writers make these awesome things that make us think about a better world. They make us think about weird, impacts of technology on society, right? Like I talked about Elysium earlier in a movie about like, we're getting farther and farther, farther and farther away from this idea that, death could be something that we could solve, right? And so it's kind of interesting as you start to look at like all of the longevity, escape velocity work that's happening. So you could get a world where you have part of the population that wants to live forever and part of the population that doesn't. And so I think some of these movies not only inspire us, but they make us start to think about like the socioeconomic or geopolitical issues that could come from some of these tech.
And out of curiosity, did you read the book Jurassic Park?
I did. And here's a fun fact about the book. Many people ask if Jurassic Park inspired George around this idea, and George will happily say he thinks that he inspired Jurassic Park, jokingly. But there is a page where there is in the first part of the book where there's actually a DNA sequence. It's exactly George's work with only one letter off. So statistically, that's probably pretty hard. And it was from published papers that George put out in the 70s. So
that would be something Michael Crichton would do, I feel like. And actually, the reason I also wanted to mention that is in the book, the Henry Wu character, he has an interesting observation about how in that story they are using frog DNA and other amphibians to fill in the gaps of dinosaur DNA. And he thinks to himself, well, these aren't actually dinosaurs because they would look quite different. I'm curious if you are able to successfully edit the genes of an Asian elephant by 2028 and we're able to recreate the features of a woolly mammoth, would it be the same species of animal that died out 11,000+ years ago?
Yeah, we get this question a lot, and it typically is after everyone's exhausted all the other debates they want to have with us, and then they fall into the it's not a mammoth category. We are trying, we've been very clear about this, that we're trying to do what's called functional de-extinction, right? We want to put animals back into their natural habitat and help the ecosystem, and we want to de-extinct core biodiversity that's been lost in traits, right? So it's functional de-extinction. It is not possible to clone an extinct species. You need a living cell, and there's just no living cells from that long ago, right? So I think it's important that people understand the difference between what's possible and what's not. But another thing that's pretty interesting is that most people don't understand speciation. Like, have you ever done 23andMe?
Yes.
Okay. What percentage Neanderthal are you?
I do not remember offhand.
But were you 0% or some percent?
Some percent.
So would you say that you're a hybrid, or do you say you're a Neanderthal, a hybrid with a Homo sapien, or do you say that you're an evolved Neanderthal, or do you say, because they did come first, or do you say that you're a Homo sapien or a human?
I say I'm Scott-Irish with a little bit of English in there.
Yeah. So I'm going to lean towards the last part of it then. So, this is what we as humans love to put things in boxes. Like the entire taxonomy and the entire way that we think about speciation, we think of it as rocks, and we're like, oh, this is a zebra, and this is a giraffe. We have to put things in boxes. That's how our brain compartmentalizes and can understand things. But speciation is more like a river, right? So all of these species evolve from each other, and we're all hybrids of a hybrid of a hybrid. And so I wouldn't say it's correct to classify our mammoth as a mammoth or an Asian elephant or a hybrid, right? And I wouldn't even classify it as a new species, right? Because there's all this debate on how speciation and our understanding of the phylogenetic tree is going to change over time. I'd really just classify it as a functional mammoth, right? And so it kind of isn't, it isn't a cold adapted, a mammoth allele adapted, edited Asian elephant, which is what some people love to say about our work. We think that if it solves the functional aspects of a mammoth, it has ancient DNA from a mammoth, and it has the lost genes of a mammoth, we just call it a mammoth. If people want to call it Mammoth 2.0, they can, or if people want to say 40 words to describe it, they can too.
Well, at the same time, you mentioned the halo effect earlier, but what advantages would we have in reintroducing a mammoth into the natural habitat of, let's say, the tundra?
Yeah, so the tundra is this completely degraded ecosystem. And we know that when we take out a species from the tundra, or from any ecosystem, whether that's a keystone predator like wolves in Yellowstone or thylacines in Tasmania, or a large megafauna like the mammoth or other elephants in Africa, they have a huge kind of cascading or tropic downgrading effect on that environment. And so what they've done is there's been these peer-reviewed papers that show that the reintroduction of cold-tolerant megafauna and more biodiversity back in the tundra actually creates this proliferation of all these additional species and both flora and fauna, because this cascading effect of seed dispersal, defecation, herding, trampling the permafrost snow in the winters. So it can actually even keep the ground temperatures colder in the summer months. So there's this weird halo effect that we've seen in these isolated populations when we've reintroduced cold-tolerant species that are much smaller than mammoths back into the Arctic already.
Would there be any concern that it could be, if it's like a creature that's been gone for almost 12,000 years, could it become an invasive species?
It's all about measuring intended versus unintended consequences. So we're working, the rewilding process is one that's like really measured. Times people are sometimes like, oh, you're gonna make mammoths and you're just gonna open the gates and like let them run free. It's a very measured process, right? So like when they put wolves back in Yellowstone or bears back in different parts of Europe or different marsupials in Australia, it's a very thoughtful, measured process with indigenous people groups, ecologists, private landowners. We've actually started putting out papers around this, and we even study things like migratory patterns for caribou, right? So we don't want to put mammoths in an area where indigenous people don't want them, where they would disrupt migratory patterns of other species. We are really looking to identify these areas that could have the best impact on the ecology, the best for the mammoths, without causing unintended consequences. The other thing that's great is they're thousands of pounds, so it's you can really measure the unintended or intended consequences pretty well.
Besides being adorable, are there any advantages of continuing the woolly mice and letting them proliferate?
So we're not letting them breed. We're not selling them, even though we get so many kids and parents calling us, begging us to sell them. We're not doing that. I do think other than the cold-tolerant experiments, the other thing that we'll probably do is if there are additional traits that we want to test from our Asian elephant editing work in Woolly Mouse 2.0, we'll probably do that.
We've been talking a lot about, obviously, the woolly mammoth, woolly mice. Do you see yourself or companies like you using this technology to save species that are on the brink of extinction right now?
Yes, and most people just want to talk about the mammoth, and I guess now they want to talk about the woolly mouse. But we actually have found that all the tech that we've started to develop, there's been a hunger for it from the conservation community. We're going to lose up to 50% of all biodiversity between now and 2050 if we don't do anything about it. Conservation works, it just doesn't work at the speed of which we're eradicating the planet and changing eradicating species and changing the planet. So for us, we've made a mandate that any tech that we make on the path to de-extinction, we open source for the world. So anybody can use it. We have 48 conservation partners around the world. Some of our early work includes a herpes virus in elephants, which you probably haven't heard of. This herpes virus in elephants actually kills 20% of elephants every year. Way more than poaching, way more than human-elephant conflict. And I'm happy to say that with our research partner, Dr. Paul Ling, only at Baylor College of Medicine, after only 2.5 years of working on it together, we now have vaccines being tested in elephants, and it's conferring resistance around the strongest strain of EEHV that kills the most amount of elephants. And so that's a tech that when we looked at the mammoth genome, we saw that they were susceptible to EEHV, right? And because they're so closely related to Asian elephants. And so with that, we said, well, we should help cure EEHV because we don't want our mammoths to be susceptible. And then we should just open source that for the world. And the success of that project has been so much that we've been able to actually start a foundation, and we raised $50 million for the Colossal Foundation to not only give our tech away, but also subsidize great people like Dr. Paul Ling on this work. So we need new tech in conservation, and we need it now, and we think this is a pretty fun path to do it.
Beyond all of this, what are the implications you think that this technology is posing that we're only beginning to consider right now?
I think that we are literally cracking the door into what's possible with genome engineering. Really kind of the combination of compute or access to compute, AI automation, and genome engineering. We're now delivering precision edits. We announced last year we did 300 edits in a fat-tailed Dunart, which is the closest living relative to the thylacine. We haven't taken a term yet, so I wouldn't say it's the most genetically modified organism yet. But we are now being able to predict things, make edits, and then get those phenotypes or physical attributes out with near 100% efficiency. That's going to change, I think, everything. I think it's going to have massive application to agriculture, human healthcare, and longevity, and even how we interact with the environment. So we launched a company last year called Breaking, and we actually took a microbe and that was found in the Amazon and supercharged it using synthetic biology, and it eats every kind of plastic that you can throw at it, right? And so that could be one of hopefully many solutions to combat the existential threat that is plastic accumulation, right? And so I think synthetic biology and these tools together can solve a lot of problems.
Well, you mentioned Peter Jackson earlier, and I know you have another filmmaker you're working with. Can you say anything about working with Doherty?
Mike is the best. Like, if you know, I mean, he's just the absolute best. He's become one of my best friends. He's awesome. One of the things we're showing at South by tomorrow is Mike actually directed a woolly mouse video. So it's going to be exactly what you think, probably. It's very short, but it's great. And Mike's gotten more and more involved with the company because he's not only fascinated by genome engineering and conservation, but how do we educate people? And we've had this like hunger from kids. And so if you can take like the Peter Jacksons of the world or the Mike Dohertys of the world and have them build educational content and inspiring content that gets kids and parents excited, we think that's pretty awesome.
Well, I haven't seen the video yet, but I hope Claire de Lune's playing.
Yeah, you'll see it. And you're like, Mike did that.
Maybe get Spielberg to come by the labs next, and that would just be full circle.
We did talk to Katzenberg in the early days, but we were like, hey, we're some random dudes that we're going to work on Mammoth, and I don't think you wanted to chat with us. But we have lately had this like influx of people from the creative world that have been pretty excited about it. We haven't talked to Spielberg yet. We haven't talked to Favreau and some of these other incredible icons yet. But we've had lots of other celebrities that have come to us saying, how can we be more involved? That's how I got to meet Joe, actually.
I'm going to guess that after this week, life will find a way in this regard.
Yeah.
Thank you so much for coming by, Ben. This was a lot of fun, and congratulations.
Yeah, thanks so much for having me.